And the subject code is PML5001, and currently we are in the Unit 3. Unit 3 is all about basics in virology. So coming to the virology, we have gone through a very long discussions. So we discussed about the classification of viruses, then we discussed about various properties of viruses. So I will do a quick revision. So we know the classification, we know their properties, we know their morphology. A rudimentary ideology about its replication. So then one thing I forgot to discuss, students, in the morphology. So I hope you already know what are these DNA viruses and RNA viruses, and depending upon their symmetry, tell me the types of viruses according to the symmetry. How many symmetries we have to the viruses? Tell me the symmetries of viruses.
Three types, sir. Three
Yes.
types, sir.
Yeah, what are the three types?
Icosahedral,
Icosahedral,
helical, and complex.
helical, and complex. Very good. And
Yes, sir.
depending upon the shape.
Sir, depending upon
Shape.
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 Bullet
Four
shape.
types, sir.
Yes.
Broad,
Bullet shape, brick shape, broad, spherical.
Bullet shape.
Yes, sir.
Okay, very good. Then how many facets and corners present to a icosahedron? How many facets and corners present to a icosahedron?
I think 20 facets.
Very good.
And 12 corners.
12 corners. Very good. Then what are capsomers? What is the location of capsomers? What are capsomers? What are capsomers? Where do capsomers present? What is the location of capsomers?
Capsid, on capsid.
Yes. On the surface of capsid, you will see capsomers. Very good. Then what about peplomers?
On the surface of envelope, sir.
Very good. On the surface of outer membrane, envelope membrane. So depending upon the presence of membrane, how we divided the viruses? Depending upon the membrane, how we divided the viruses? We have two types. Can anyone say?
Sir, envelope and non-envelope.
Enveloped viruses and non-enveloped or naked viruses. Very good. Then what is the average size of the virus?
20 nanometer to 250 or
250 to 300 nanometers. Very
300
good.
nanometers.
Very good. Then, okay, this is fine. Now, students, okay, I have given the names here. Anyway, now tell me, students, I will point out one virus and you need to tell its properties. This one. Tell me the properties of this virus. That means you need to tell it is icosahedron, enveloped icosahedron, DNA virus. Okay. Similarly, you need to tell all the viruses. What type of virus is this?
Sir, this one is non-envelope virus.
Naked
virus. With icosahedral.
Icosahedron, non-enveloped virus. So symmetry is icosahedron and non-enveloped icosahedron virus. Very good. Then what about this one? What type of virus is this? What type of symmetry is this? Is it icosahedron or helical?
Helical, sir.
Helical.
Helical, sir.
It is a helical. Is it enveloped or non-enveloped virus?
And enveloped virus.
Enveloped. So paramyxo is a helical enveloped virus. Okay. Then what about this one? Bunya virus. Tell me this virus. It's a helical enveloped virus.
Yes.
Helical enveloped virus. Then this one. What type of virus is this? Tell me its symmetry, morphology, shape, everything.
Sir, bullet shape.
Bullet shaped, very good.
Envelope.
Enveloped, very good.
And helical, sir.
Helical, very good. Symmetry is helical, it contains envelope, and it is bullet shaped virus, which is the rabies virus or rhabdovirus. Very good. Then what about this virus, students? Corona, the recent celebrity, is it enveloped or non-enveloped?
Enveloped, sir.
It is
And
enveloped helical RNA virus. Okay, Corona is a enveloped helical RNA virus. So now you are getting some idea about viruses. Now you can say precisely, if someone asking what is coronavirus means, you can say precisely, it is a helical enveloped RNA virus. So now you are getting some idea, right? What are viruses and their morphology? Right, students?
Yes, sir.
So yes, so very good. So now you got some idea about all the viruses, and kindly try to draw individual morphology of all the viruses. Try to give some important viruses. At least you need to memorize pox virus morphology, herpes virus, paramyxoid, adenovirus, and also rabies. Okay, and corona also. So in the coronavirus, on the envelope, you can see crown-like structures. So presence of these spike proteins makes it is a unique feature for crowned or coronaviruses. Okay, then in the herpes virus, along with this envelope, all herpes viridae viruses, that means alpha, beta, gamma, herpes simplex 1, 2, varicella zoster, cytomegalovirus, and Epstein-Barr virus, all herpes viridae viruses have a extra outer membrane. Just beneath the envelope of these herpes viruses, you will find a second membrane called tegument. This membrane is called tegumentum membrane. So presence of tegument membrane is a characteristic feature to identify herpes viruses. So this tegument membrane will give additional protection to the virus. So this tegument membrane is resistant to many factors within the body. So virus can able to resist all types of immunological attacks that are doing by the body. So because of this tegument membrane, these viruses are highly specialized at causing carcinomas. That means they can cause cancers in the body. So all herpes viruses have the tendency to develop cancers in the body. Okay, so this is a few interesting points about viruses. Then we discuss the classification. Then we discuss the short introduction. Then we discussed about morphology. Morphology we break down into morphology, chemical properties, and the susceptibilities. In the morphology, we discussed about their size, shape, symmetry, their nuclear properties, chemical properties like nucleic acid, proteins, lipids, and we even discussed about viral susceptibilities like temperature resistance, pH resistance, lipid solvents, disinfectants, and radiation resistance. So this is all about the morphology of the viruses. Then we even I have given an idea about how to draw the icosahedron also. So this is the basic structure of icosahedron with 20 facets and 12 corners. Right? Then let us move on to the next topic. So kindly start writing notes. So the next topic is viral replication or replication of viruses. So in today's lecture, we will discuss about replication of viruses. And in the afternoon session, I will discuss the next topic. So today I will discuss replication of viruses. Okay. So is everyone following me? Students, are you following?
Yes, sir.
yes, sir.
Perfect. So let me see the attendance here. 38. Perfect. It's a good number. Coming to the replication of viruses, we already know basics of replication of virus. So first virus need to attach to the cell surface, and this attachment of virus to the cell is called adsorption or attachment. After attachment, virus will penetrate into the cell. This is called penetration. After penetration of virus into the cell, now the virus will uncoat its proteins. It will uncoat its outer shell. It will uncoat its either envelope or capsid. So that stage of virus is called uncoating. After uncoating viral proteins, the virus will start biosynthesizing the viral proteins. The viral RNA or DNA will start biosynthesizing in the cell. So the ribosomes will start synthesizing the proteins of viral particles. Yes, sir. Hour 15 and I sent you one more PDF of CET. Both I need, sir, print. Sir, I have limited papers. This is my final result. Can you see? One set is fine, sir. Just. Thank you. Sorry, students. Coming to the point. So viral replication. So first virus will attach, then it will penetrate, then it will uncoat its viral capsid, then it will start biosynthesizing the RNA or DNA. Okay, so after biosynthesizing, these proteins will assemble together to form a baby virus. That assembly of proteins is called maturation. So the assembly of viral proteins within the cell is called maturation. So after maturing the virus, now this virus will be released out in two forms. One way, virus will come outside in a form of budding. That means virus will acquire a tiny portion of the cell membrane, and such release of virus is called budding. So via budding, we will get enveloped viruses. Whereas non-enveloped viruses, they will destroy the cell membrane and they will come outside. And that is called lytic form of coming out. So these are the important stages in the viral replication, starting with absorption till the release of the viruses. Now,
Sir, excuse me, sir.
yes,
Can you repeat that point
which point, Eric?
on the replication the cell?
On the release of the virus.
Yeah.
See, very simple. I will draw a rudimentary picture here, just to see. So this highlighter. See, so I'm drawing two cells here, cell 1, cell 2. So in the cell 1, we got a viral attack. So this in the cell 1, we have virus. Now, when this virus coming outside, what it will do is it will push the cell membrane. When it is pushing the cell membrane, so virus came little bit outside here, and it also pushes the cell membrane gently. So due to this gentle push, virus will push the cell membrane like this. So when the virus ultimately coming outside, it will come along with the cell membrane. So when the virus coming outside the cell, so this is virus, when it is coming out, it will acquire a portion of the cell membrane around it. So we are saying enveloped viruses, virus has membrane. Actually, that membrane belongs to the living cell. That is our own cell membrane, which was coated by the virus. Such release of viruses are called budding. Via budding process, virus will acquire membrane. This membrane is actually cell membrane only. It is taking cell membrane as its protective covering. So such release of viruses is called budding. Okay, so budding of viruses. And second one is in the second form, viruses will be very aggressive. So virus replication will be too high. There will be some 1 lakh to 2 lakhs viruses in a single cell. Because of this overload of viruses within the cell will start expanding, cell will start bulging like this. So first cell will go hypertrophy, that means cell will enlarge. At one particular point, cell will burst. Cell will be sliced into tiny particles. So cell will burst, then viruses will be released out. So this is a very deadly form of virus release. Okay, and this form of release is called lytic phase. Lytic release of the viruses. LYTIC. Lytic means to break. So these viruses will break the cell. If that is the case, these cells will go immediate necrosis. These cells will undergo unrecoverable or unrepairable damage. It will cause unrepairable damage, permanent damage to the cells, and we cannot retain such cells. So one best example is cirrhosis, liver cirrhosis. Hepatitis virus will cause irreversible damage to the cells of the liver. So if you get hepatitis means you can say goodbye to your liver. Very, very deadliest form of viral infection. So this is how the viruses will released from the cells. Okay, so these are the 6 stages of viral replication, starting with the attachment, penetration, uncoating, biosynthesizing viral proteins, maturation of viral proteins, and release of viruses. Okay, so these are the 6 stages. Now, I will add one important point. Kindly pay attention. See, students, when the virus replicating inside the cell, it is hijacking the cellular normal machinery. It is hijacking the cellular ribosomes. What is the purpose of ribosomes? Ribosomes will prepare proteins for the cell. Now, the virus using these cellular ribosomes for preparation of viral proteins. Instead of cellular proteins, now the ribosomes will start manufacturing the viral proteins. That means it is shutting down the normal metabolic activity of the cell. A virus will shut down the normal metabolic activity of the cell and it will divert the metabolism of the cell for its replication, for its development. During this, when the ribosomes preparing the viral proteins, these ribosomes will also prepare waste products. Waste cellular proteins will be generated by the cell that is infected by a virus. I will give you a small idea here. Imagine we are making a ice cream. So when we are making an ice cream, we will we need to cut the ice cube into a particular shape. First, we will get a big ice cube and we need to cut the ice into small cubes. During this time, we may waste some ice. We may waste some ice or some plastic debris. That wasted plastic debris, here in case of cells, those debris will be protein debris. Such protein debris are called inclusion bodies. These are the leftover proteins by a virus. When the virus replicating inside the cell, it will leave some waste protein products. These leftover waste protein products are called inclusion bodies. So these are literally, we can say, virus excreta. We can say viral waste products. These viral waste products are called inclusion bodies. Now, there is a advantage of these inclusion bodies. The advantage is each virus will have its own specific inclusion bodies. The shape of these inclusion bodies will be specific to a specific virus. I will give you one idea about inclusion bodies. Here, we are identifying virus indirectly by its waste products. That means, imagine you have a goat, you have a cow, and you have another animal. We know that goat excreta will be like a small black balls. So goats will excrete like a small black balls. So if you see small black balls, you can indirectly say, yes, there is a presence of goat in this area. Or if you see a large lump, green lump, then you can say might be some cow or buffalo roaming around this area. So we don't know whether cow or goats present there or not, but by their excreta, we can have a indirect assumption that these particular animals may roam here. So by seeing their waste products, we can able to guess. Similarly here, we will guess the viral presence by detecting the inclusion bodies present in the cell. Another advantage of inclusion bodies is inclusion bodies are comparatively larger than viruses. We cannot visualize virus in the microscope, but we can able to visualize the inclusion body under the microscope. So detection of inclusion bodies will be less hectic or easier task to identify virus rather than identifying the actual virus. So we have two types of inclusion bodies. I will tell. One type of inclusion body is RNA inclusion bodies. RNA inclusion bodies means RNA viruses will replicate in the cytoplasm of the cell. Keep in mind, I'm telling again, RNA viruses will replicate in the cytoplasm of the cell because RNA viruses already have RNA and RNA can be directly introduced into the ribosome. Ribosome already present in the cytoplasm. So the ribosomes will start preparing the viral proteins. So RNA viruses will replicate in the cytoplasm of the cell, whereas DNA viruses will replicate in the nucleus of the cell, because DNA viruses must introduce their DNA into the nucleus of the cell. Obviously, DNA viruses will replicate inside the nucleus of the cell. So if you see inclusion bodies in the nucleus of the cell, then we can assume that it is a DNA virus, because they are nuclear inclusion bodies. Nuclear inclusion bodies can be developed only by DNA viruses, whereas cytoplasmic inclusion bodies will be developed only by RNA viruses. RNA viruses will develop cytoplasmic inclusion bodies, whereas DNA viruses will develop nuclear inclusion bodies. So now I have given a point on inclusion bodies. Inclusion bodies are the viral leftover protein products that has been left within the cell. These can be either DNA nuclear inclusion bodies, or these can be cytoplasmic inclusion bodies. Furthermore, these inclusion bodies are further classified, that I will discuss later. So as of now, just remember, during the replication of virus within the cell, virus will leave certain leftover proteins. These leftover proteins within the cell are called as inclusion bodies. These can be either nuclear inclusion bodies or cytoplasmic inclusion bodies. Cytoplasmic inclusion bodies by RNA viruses, whereas nuclear inclusion bodies by DNA viruses. Is it clear for everyone so far? Are you listening me? Is everything clear so far, students?
Yes, sir.
Sir,
Yes. Yes, Eric?
sir, is all types of viruses rupture the cells, or some viruses rupture the cells?
Only some viruses. Not all viruses will rupture the cell, but all viruses will cause cell damage.
Okay.
All
And
viruses will cause cell damage. Yes, yes, Eric.
sir, do we have vaccines for DNA viruses?
Yes, we do have. We have very efficient vaccines for both DNA and RNA viruses. The best
How
vaccine
the
is
D-
hepatitis B vaccine.
okay, how the DNA vaccine will work on the cell?
How the DNA? Okay. Uh, See how the vaccines will work is in the vaccines. Forgot about either it's a DNA or RNA. Just keep it's a virus. You just keep in your mind it is just a virus. Now, whenever virus entered into our body, as a whole, the immune system will get recognized. It will get alerted, and immune system will develop antibodies, and these antibodies will kill the viruses. So the way vaccines work is in the vaccines, either it is a DNA or RNA vaccine, or simply DNA virus vaccine or RNA virus vaccine. When this viral particle introduced into the body immune system will get activated and it will develop antibodies. So development of antibodies is the key property in vaccination. So rather than giving a live virus into the body, here we will give killed virus. So this killed virus also looks like a real virus, and immune system will get recognized and it will develop antibodies towards the virus. So that is how the viruses, the vaccines will work. So you need not to worry about either it's a DNA or RNA vaccines. Both can trigger immunity in the body. So that will be at extracellular level. So in virology, keep in mind, students, vaccines work at extracellular. That means it's a collective uh, technique. So whole immune system will work to kill the viruses. That is immunity. Immunity is entirely different from individual cell machinery. Now, still that is a very good question, Eric. Remember, whenever the virus entering inside the cell, virus will hide from the immune system, because now the virus present inside the cell and immune system cannot recognize something which is beyond inside the cell. So if any helper T cell or if any macrophage wandering near the infected cell, it cannot recognize it, because the virus is inside the cell and it will think cell is fine, but cell is under attack, cell has been hijacked. But that information is not known to this immune cell. but we still have a cellular level immunity in the body. Even these cells, the infected cells, will try to destroy the virus. Even the infected cells will try to destroy the virus. That can be achieved by releasing a viral specific cytokines. Those cytokines are called interferons. Whenever virus enter inside the cell and it is trying to manipulate the DNA or RNA of the cell will immediately release a snipping proteins, a cutting proteins called interferons. Interferons are the antiviral proteins that are released by an infected cell. So it's a very clever strategy. In the way interferons work is really clever because these interferons always guide. They will patrol the normal DNA. They will check all the DNA in the cell. If there is any suspicious in the DNA, immediately they will cut the suspicious DNA portion. So they will guard the entire DNA of the cell. So they will guard the DNA and RNA of the cell. So if they suspect anything wrong, they will immediately chop. Okay. Actually, they're called chap aprons. I'm not going up to the molecular level virology. We can have a generalized topic, okay? But still, we have a individual cellular immune mechanism. That cellular immunity is mediated by interferons. Okay, so interferons will clear the cellular DNA or cellular viruses. But these viruses are so smart that they will escape from this interferon activity also. They know how to escape from interferons activity. Pathogens are really clever. They have evolved multiple times. Yes. Any more doubts?
Sir, we have heard about mRNA virus.
Yes, mRNA virus.
Yes. So this mRNA virus, it means that replication of these killed virus will be occurred every day in our body, in every life, because
Yes,
synthesis of protein is going on every time.
exactly. Actually,
So
we are all. Mm, how?
how this will occur? Because mRNA virus will go to touch the protein synthesis and occur being produced together.
Yes.
So my doubt is
Mm.
these killed vaccines will always be generated as the protein being produced together.
These killer vaccines.
Because
Mm.
protein synthesis occur daily, right?
Mm.
And this mRNA vaccine go to touch the protein synthesis.
Perfect. I got it. Okay. How mRNA vaccines work? So
Yeah.
got it. So how mRNA vaccines work is just remember, students, how the immune system activated. Whenever we got a virus, it helper T cell first macrophage will digest the virus. After digestion of virus by the macrophage, it will express the viral proteins on the surface of macrophage. Then that is called antigen presentation. Now this antigen will be read by a nearby helper T cell. Now this helper T cell will go to 1 and 2. Helper T cell 2 will go to the nearby B cells, and those B cells will start producing viral specific antibodies. How the B cells producing viral specific antibodies is my question. Okay, listen. Now, a B cell will take the protein information from a helper T cell. So from helper T cell to a B cell will get the protein information. So it will say so and so protein. So and so protein containing virus entered into body and kindly prepare antibodies for it. Now, this B cell need to develop similar antibody. To develop similar antibody, B cell need to get a information. B cell DNA need to get information to develop the protein, a specific protein. That means it needs to work out on its DNA level. Now it need to change the DNA in instructions. It need to prepare an instructions to prepare a protein for the virus. To prepare the instructions, it must prepare a DNA. Then this DNA need to be converted to RNA, and this RNA must code into a specific protein, and this protein, that means here it is antibody. This antibody can kill a virus. Now, In a basic killed virus vaccine, we need to go with this elaborated pathway. First, the killed virus should be digested by macrophage. It need to be expressed by the helper T cell, and that need to be get activated by the B cell. And B cell must recognize the virus, and it will take 1 week to develop antibodies against a killed vaccine. To speed up this procedure, why can't we introduce direct RNA, direct viral RNA into a B cell? So we will give direct information to the B cell. So directly we will target B cell and we will give, this is the protein, this is the viral RNA, and I need antibody for this particular RNA. And B cell will get immediate activation and it will start producing antibodies immediately. So the time duration for a normal vaccine to work will be 2 weeks. But the time duration for RNA vaccine will be within 1 to 2 days. So within 1 to 2 days, the B cell will produce enough antibodies to develop antibodies. So that is how the DNA and RNA vaccines work. So here in DNA and RNA virus vaccines, we will directly give the viral RNA or DNA directly to the B cell. So B cell can immediately recognize it. But the problem is it need the way we will administer these drugs is highly cost effective. So to develop such vaccines, they are very costly. So first we need to understand the complete sequence of genome of virus, really costly affair. So that is why we will go with this killed vaccine technique. Even though late, but still it will give the same amount of immunity, but lately. But a healthy population, they can get immunity via killed. But RNA and DNA vaccines are more efficient than killed. I'm agreeing with it. So that is how RNA and DNA vaccines will work. You got my point, right, Eric?
Yes, I got it.
Yeah. Actually, virology is really interesting subject. So here we are, we are literally manipulating the cell. So first we understood how the cell working or the life, and now we are playing with the life. It's a so there is a war between two people, and now we are intervening in the war, and we are trying to justify it. It's a really interesting subject actually. So coming to the viral replication. So first one is attachment. So what is attachment? Attachment means virus will attach to the cell. Now listen, students, very important. Each and every cell of our body, each and every cell of our body have cellular receptors. Their own specific receptors present to each and every cell. Now we are coming to the level of cell, and now we are discussing only the cell properties. Listen. Now, if it is, I don't have any pen here. Okay, imagine if it is a pancreas cell. On the pancreas cell, it will have some sharp tooth-like receptors. Okay. If it is a mast cell, then it will have some fiber receptors. Similarly, each and every cell has receptors. Okay, or proteins, surface proteins. Believe it or not, each cell will have around 20 to 70,000 receptors. There are totally 70,000 receptors for a single cell. 70,000 individual receptors are present. I think it is way more high than 70,000. Okay, 70,000 receptors to a single cell. Sandeep, I'm getting noise from. Okay, he left. Fine. Now, let me give you one particular cell receptor, which is CD4 helper T cell. Helper T cell will have a receptor called CD4 receptors. CD4 receptors present on the surface of helper T cell. Okay. Now, viruses are evolved to bind to this CD4 receptor. Such virus is HIV virus. HIV virus can able to bind to the CD4 receptor along with a co-receptor called CXCR5. CXR and CXCR5 are the co-receptors of CD4. These two receptors are very important for a HIV virus to attach onto the cell surface of a helper T cell. So HIV virus will attach itself to the helper T cell via these receptors. These receptors are called as CD4 and CXCR4 receptors. Okay, receptors present to the cell, whereas virus also contains a hand-like structures. These viral attaching structures are called ligands. The receptors present to the virus are called ligands, whereas receptors present on the cell are called receptors. One best example is HIV virus. HIV virus have a ligands that can able to specifically attach to the surface of CD4 cells. Okay, so HIV have a ligand called GP120. Glycoprotein 120 is a ligand present on the surface of HIV virus. So this my fist is a HIV virus and it has a ligand called GP120, and this GP120 can able to bind to the CD4 receptor of the helper T cell. So with the help of this, it can able to attach itself to the cell surface. So this attachment happens via ligands of the virus and receptors of the cell. So cell receptors and ligands of the virus work together for the attachment. So that is why each virus is adopted to attach to a specific cell of our body or to a specific receptor of our body. Hepatitis viruses have ligands that can attach to the liver. HIV viruses have ligands that will attach to the helper T cell. So viruses are always cellular specific in nature. They are highly specific in their targeted cells. Liver viruses only attacks liver. Brain viruses only attack brain. Immune viruses only attack immune cells. So the reason is because of this specificity in the ligands and cellular receptors. So this is all about the attachment. So whenever the virus come in contact with the cells by random collision, but absorption or attachment is mediated by binding of viral surface structures called ligands. So these ligands will attach to the receptors of the cell surface. In case of influenza virus has hemagglutinin ligands. These hemagglutinin ligands will attach to the red blood cells and the mucous membranes of the lungs, and they will cause the infection. Okay, or HIV viruses. HIV viruses have GP120 ligand, which will bind to the CD4 receptor of the helper T cells. So this is all about the attachment of viruses. Is it clear, students?
Yes, sir.
Okay, then next is penetration. So penetration, there is nothing much. So penetration means now the virus will penetrate into the cell. Okay, now this penetration can happen by two forms. One is by phagocytosis. So phagocytosis means cell itself will try to engulf the virus, and that is what the virus needs. Usually what cells will do? Cells will try to do phagocytosis. They will try to engulf the virus. The same, actually cells don't know that they're intaking a toxic agent. So they're thinking that they will digest the pathogen, but pathogen needs this engulfment. So it's a very clever strategy of viruses. And engulfment of viruses by cells is called viroflexis. So viropexis is a phenomenon of engulfment of phagocytosis of viruses called viropexis simply. Okay, so whereas enveloped viruses, how enveloped virus enter into the cell means they will fuse the cell membrane to the cellular cell membrane. That's it. Enveloped viruses will fuse their envelope into the cell membrane. So due to that, the membrane will fuse and only the viral capsid will enter into the cell. So this is how the virus will penetrate into the cell. After penetrating, now virus will remove the coat of the capsid, and removing of this outer coat of capsid is called as uncoating. So uncoating means this is a process of stripping the virus outer membranes. So the viral nucleic acid or DNA will be released into the cell. So this particular stage is called as uncoating. So the third stage is uncoating. So in the uncoating, the viral nucleic acid will be destroyed. After this, there is another interesting thing about uncoating. So uncoating will be done by cellular enzymes only. So the enzymes present in the cell, the same enzymes will help for the virus to remove its coating. So we ourselves removing the cloths of a warrior to see his gun or his weapons. So the cell itself helping the enemy to do its job. Okay, so uncoating is done by the cell itself. Cellular enzymes themselves will remove the viral proteins. Okay, so uncoating. Then after uncoating, biosynthesis will happen. So in the biosynthesis, nothing much, students. So DNA viruses will replicate in the DNA in the nucleus of the cell, whereas the RNA viruses will replicate in the cytoplasm of the cell. Okay, that's it. That is the general statement. Whatever they're saying here is same thing. DNA viruses will replicate in the nucleus of the cell, whereas RNA viruses will replicate in the cytoplasm of the cell. Okay. Next, after biosynthesis, what is that? Maturation of the proteins. Okay. So here in the biosynthesis, I have given some more things like mRNA transcription, DNA transcription. This is not required for you. Okay. So this is molecular level. This I'm just ignoring it. Okay. Then maturation. So after synthesizing the viral proteins, these viral proteins will assemble together to form a baby viruses. And this assembly of viral proteins is called maturation of virus. Okay. So the viral nucleic acid polycapsid, viral capsid, virus, capsomers, viral envelope, viral everything, viral all viral proteins and viral DNA or RNA will be matured. They will assemble within the cytoplasm of the cell. Okay, and this is called maturation of the virus. After maturation, the virus will be released from the cell. This release will happen by two methods. It can be by budding or it will be by lytic phase. So budding means the virus will move outside along with the cellular membrane, that is called budding release of the virus. And some viruses will destroy the cell membrane, and that is called lytic of the lytic release of the cells. Okay, now one important point I need to discuss, students, about eclipse phase. What is this eclipse phase? See, listen, very important point. Now I will discuss about eclipse phase. See, when the virus is outside the cell, our immune system can detect it. Our immune system antibodies or helper T cells can able to detect the virus. Okay, so our helper T cells can able to detect the virus when the virus is outside the cell. But when the virus entered into the cell, when the virus penetrated into the cell, from the stage of penetration till the release of baby viruses, maybe it will take some 20 hours. Let us assume it is 20 hours. From the penetration of virus till the release of virus, the virus will spend its time within the cell. During these 20 hours, our immune system will be clueless. Our immune system don't know where is the virus. Virus will hide within the cell at this time. Our immune system don't know where is the virus because virus is inside the cell. Now it is replicating inside the cell. And this particular missing period is called incubation time. This incubation period is also called as eclipse phase. This incubation period is also called eclipse phase. During this eclipse phase, we cannot detect virus in a patient body. The patient will get negative test results. No matter whatever lab test you are doing, you won't get any report. You will get negative, but the virus is inside the body. Even the virus present inside the body, we won't get any results. That is because of this eclipse phase of viruses. And this eclipse phase will vary from virus to virus. In case of DNA, in case of HIV, it is almost 1 to 3 years. In case of hepatitis, it is almost 6 months. In case of coronavirus, it is almost 10 days. So first 10 days, these are the asymptomatic phase of the virus. During this first 10 days, we won't find we won't get even positive result. Okay, and this duration is called eclipse phase. Okay, so eclipse phase means from the stage of penetration of virus into the host till the appearance of first progeny viruses, the virus cannot be demonstrated in the blood samples, and this period is known as eclipse phase. The normal duration of eclipse phase can be either 15 to 30 minutes, half an hour to 2 days. So half an hour to 2 days will be the normal incubation period of the virus. This is called incubation period, or you can also say it is a eclipse phase of the virus. Eclipse phase means it is the time duration from the penetration of virus till the appearance of baby viruses from the cell. We cannot detect the virus, and this period is called as eclipse phase. Is it clear, students? Do you got idea about what is eclipse phase?
Yes, sir.
Yeah, this is
Yes,
eclipse
sir.
phase of the virus. Thank you very much, sir. Thank you. So you took prints of both, right? Thank you thank you very much. Okay. Yeah. So that's it, students. So coming this slide, I think I need not to discuss this with you. As I said, we have DNA and RNA viruses. DNA also we have double-stranded DNA viruses, single-stranded DNA viruses, positive stranded viruses, and negative stranded viruses. So if you want an idea about what virus is what, then you can have a look at this slide. For example, adeno and herpes and pox viruses are double-stranded DNA viruses, whereas single-stranded DNA virus is parvoviruses. Double-stranded RNA viruses are reoviruses. Positive single-stranded RNA is picorna. Orthomyxoviruses are negative single-stranded viruses. So these are the classification of viruses at their genetic level. At their genetic level, this is how the viruses are classified, and this is the true classification of viruses. This was given by David Baltimore. Okay, for this he was awarded Nobel Prize for precisely classifying the viruses depending upon their molecular level, depending upon their synthesis of RNA. Ultimately, no matter whatever the virus is, ultimately it need to generate a mRNA. How the mRNA is generated? By seeing the way it is generating mRNA, we have classified these viruses. We have double-stranded DNA virus. Double-stranded DNA virus can able to generate mRNA within a single step, whereas single-stranded DNA virus will convert into double-stranded to form a mRNA. Then all positive or negative stranded, then they need to go via phase. Actually, I forgot this this molecular mechanism, but just have an idea. Okay, so this is how the virus is classified depending upon the molecular basis. So a quick revision of replication of virus. So viral replication happens in 6 stages: absorption, penetration, uncoating, biosynthesis, maturation, and release. So absorption is mediated by ligands and cellular receptors. Penetration happens by phagocytosis. Can anyone say the viral phagocytosis name? It is called as viropexis. Okay, viropexis is phagocytosis of virus by a cell is called viropexis. Then uncoating will happen. Uncoating means the viral proteins will be removed by the cellular enzymes. So the viral DNA or RNA will be exposed. Now this DNA RNA will go for biosynthesis. DNA viruses will replicate in the nucleus of the cell, whereas RNA viruses will replicate in the cytoplasm of the cell. Then virus proteins will be matured. Viral capsid, viral envelopes, viral genetic material will get matured within the cell. They will assemble within the cell and they will release. Release can be either of two types. They will release by budding or they will release by lytic phase. Then after releasing, viruses will go for eclipse phase. Hello, ma'am. Yes, ma'am. Yes, ma'am. Ma'am, I got all the prints, ma'am, of all the departments, CVT, MLT, and okay, ma'am. Okay, I will take the print, ma'am. Sure, ma'am. Sure. Okay, thank you. So this is all about viral replication, students. Any more doubts in viral replication? All right. So in the afternoon session, we can go for viral cultivation. Very interesting topic. In the afternoon, we will go with the viral cultivation. Okay. So thank you very much, students. Any more doubts in topic? In today's topic? Sir, I was little bit busy today.
No, sir.
So thank you very much, students. You can leave.
No, sir.
Fine. So I will see you in the afternoon. Okay. Thank you very much.
सर नेक्स्ट कब ज्वाइन करना है? ओए शाहरुख, हेलो।
अबे रूटिंग नहीं है क्या तुम्हारे पास? रूटिंग देखना कब ज्वाइन करना है।